Can it stop?
No, it never
Can
stops.
physiology doesn't understand that we're good and safe? You ready?
Ready, start yelling.
All right, folks, we're going to get started with the third symposium of the day. So go ahead and take a seat at your leisure. First, we'd like to introduce ourselves. My name is Luke Crestline. I'm currently a postdoc with Systematic Entomology Lab in Washington, D.C.
And I'm Jessica Awad. I will be starting at Naturalis Biodiversity Center next year. Well, people are still trickling in.
Yeah.
All right, shall we just start?
Yeah, let's just get
Okay. Yeah, so for this symposium, I think it's pretty clear that everybody here knows the importance of entomological collections, but institutional administration and funding agencies often don't understand the role that collections play in scientific research, or they don't place a high priority on studying biodiversity for its own sake. I think especially now, as the world is changing, we can expect even less investment in studies that deal with biodiversity monitoring and conservation, and more focus on questions like, can I eat this? Or can I sell this for a profit?
However, collections are also really important for answering these questions. And our symposium, the goal of our symposium is to highlight research that uses entomological collections to solve problems concerning agriculture, economics, and biological control. We're excited to have speakers from a diversity of academic, museum, and government collections, and to highlight their work on a diversity of taxa to solve a variety of problems. So we're going to start with our first speaker, Erin Powell, who is from the Florida State Collection of Arthropods.
Have a clicker? Or no? Not working. Okay. All right. Hi, everybody. My name is Erin Powell. I'm just finding the time on the slide. Okay. So I am a research scientist and curator of scale insects and whiteflies for the Florida State Collection of Arthropods. So one of my main duty is just identifying scale insects and whiteflies that are either already in the state of Florida or coming into the state of Florida with agricultural commodities. And today I'm going to talk about a couple case studies of kind of interesting stories of insects that have shown up in Florida and how we've used the collections to solve those. Over there. Okay. All right. So Florida is particularly susceptible to invasive species. We've got a nice warm subtropical climate. We have lots of ports of entry. We have a booming agricultural trade. So between 1990 and 2023, we had over 450 adventive species of arthropods. That's over 1 per month coming to Florida. And by and large, Hemiptera is the largest order that we're seeing as invasive pest species coming into the state. So within 12 years, we had 61% of the regulatory significant pests that were intercepted at our highway interdiction stations that belong to the order Hemiptera. So those are things such as armored scale insects on avocados. So the Florida State Collection of Arthropods, if you're not familiar with it, is one of the largest collections in the southeastern United States. We have over 12 million specimens. A lot of our samples submitted by inspectors that are looking at nurseries and interdiction stations and other imports throughout the state are, they're submitting samples to the collection, and those are being integrated into our collection. Also, the curators are, doing their own collecting efforts, and we have donations and trades going on. So ultimately, the Florida State Collection of Arthropods was started as a reference collection to protect Florida's agriculture. So specifically looking at our scale insect and whitefly collection, it's one of the top 4 in the country with over 100,000 slides. We are rivaled by the USNM Smithsonian, which is the #1 overall, but we have, comparable numbers of types and species representation with the CDFA collection and UC Davis. So why are invasive scale insects and whiteflies so incredibly important for us to be paying attention to? Scale insects are notoriously resistant to pesticides. They have rapid generation times. Sometimes they're parthenogenic, so they can just come in with a single female and start a whole new population. They may be vectors of plant viruses. They produce honeydew, which promotes the growth of sooty mold, and they just overall can reduce plant vigor and cause plant death. So scale insects are often thought of, or in whiteflies, as kind of pitiful. You know, they've got like immobile females that don't even have legs, yet they're excellent invaders. They're notorious for moving around the world rapidly. And this is mostly via the human, agricultural trade. They're so tiny that they go, unnoticed, undetected in inspections, just because we can't inspect every square inch of every plant, material that comes into the country or into the state. And then once they arrive, they move around via wind. They can just the crawlers, the first instars that are the most mobile stage, can get picked up by the wind and be moved around. And then they're also known to be phoretic on other more mobile insects. I always like to show this cool SEM image of the tarsal digitals on armored scale crawlers are like little suction cups to grab onto more mobile insects. So I'll talk a little bit about 3 case studies. An armored scale that arrived in Florida in 2022, a mystery mealybug that showed up on the roots of Biden's alba in 2018, and then an interesting whitefly that showed up on ferns in the Fairchild Botanical Garden. I believe that was in late 2021, early 2022. So first, this is just an example of, okay, well, we've intercepted it hundreds of times. Surely it's eventually going to establish itself in Florida. And that is this great example. So we have this armored scale. The USDA APHIS intercepted it over 240 times over a span of 30 years. And this one was interesting because it's an Asian in origin, but then all of a sudden the data showed that it started to shift and it was actually coming from Costa Rica. And we didn't even officially record it from Costa Rica until 2019. And then it was just casually mentioned in a molecular paper that they had gotten specimens from Costa Rica. So it wasn't surprising at all that it established. And in this case, it was nice that we had all of this reference material from these interceptions in the collection. So once it did finally establish in the landscape after all of this pressure, we could confidently say, yep, that's it. Not surprising that it has made it here. And now it is in Broward and Miami counties and expected to spread and continues to be intercepted on nursery stock. So for this project, this one was interesting because I ended up going through all of our Lepidostaphyes slides in the entire collection, found some misidentifications, realized that there was a species from Croton that had been intercepted more often than we realized in the past, reaffirmed that one of the pine species is not actually present in Florida. And looking in the collection and getting these lists is really important so that we know what is actually regulatory. If we don't know whether it's in Florida or not, we can't make regulatory decisions on whether to turn out material that has that species on it. So ended up making a key to all the species of Florida. So that'll be a nice diagnostic resource going forward. I mentioned that we ruled out parasitoids, not necessarily because it's related so much to the collection, but it's interesting because our collection can sometimes actually have parasitoids within it. Sometimes these scale insects get mounted with the parasitoid wasps within. So again, collections can help us know what's in Florida, but also what's already parasitizing the species that are coming into Florida and what can be used as useful biological controls. So if you'd like to know more about that one, that was published in Insectamundi recently. There's another mussel scale in the same genus that's on the move that we're intercepting repeatedly on Lucky Bamboo. So this is another one where we have a great amount of reference material. So when it does possibly establish in the landscape, we will have good material to compare. So in 2018, a mystery mealybug showed up on the roots of Biden's alba. And this one was a fun one because it was originally identified as Phoenococcus decembrifolium, which is a species of mealybug that was described from Uruguay. But we couldn't get our hands on the type material because the types are actually in Argentina and they couldn't be sent to the USNM where they were supposed to go. So this thing started rapidly spreading onto lots of different roadside weeds. Then it was jumping onto tomatoes and eggplants and other solanaceous crops. So raised our hairs. We wanted to look into it a little bit more. And it turns out that a species very similar to Decembrifolium maruku was described from Japan in 2022 after we had done our original identification of this in 2018. So we ended up sequencing it, comparing it to this Japanese species, and it turns out that it's a match to this. And lo and behold, the Japanese thing also presumed adventive from the neotropics. And what did it show up on? Biden's pilosa, which is a synonym essentially of Biden's alba. So this one was a fun little one where it's like, well, we didn't have any reference material in the collection. It was definitely new to Florida. We scoured every Phoenococcus slide in the collection. It definitely, was only showing up in 2018 for the first time, and couldn't really put a name on it just because the taxonomy hadn't advanced enough. It's always possible that Cimbrifolium and Maruku are still synonyms, but again, we really need to see the type material from that's in Argentina from Uruguay. Again, if you're interested in this one, this was just recently published in Florida Entomologist. We also did a key to all the species of Phoenococcus in Florida for a diagnostic resource in the future. Third example, this is my favorite. We had an export inspection of endangered ferns that had been propagated at the Fairchild Botanical Garden as part of a conservation program. And they thought that, they were clean, they were going to send them back to Puerto Rico to this native plant nursery. Well, lo and behold, they have these tiny, tiny little whiteflies on them. And we were like, well, we really need to look into this more before you go off and send this highly endangered fern back to Puerto Rico for this whitefly to jump onto the, the last 7 plants in the wild. So this one was really interesting because whiteflies don't really like ferns. Only 14 species, less than 1% of all described whiteflies actually feed on ferns. So it was already interesting in that regard. It was also interesting because it actually stacks its cuticles, which turns out a lot of whiteflies do this, but it's not really talked about in the literature about like why or how. Presumably could be an anti-parasitoid thing if you've got all of your instars stacked on top of each other. So you're looking at a 4th instar pupa, a nymph, for this whitefly. And you can see in the slide-mounted specimen, these operculum, the lingula, that's a part of the whitefly pupa that you can kind of tell that it's a sandwich of 4 instars that have all been stacked on top of each other. So we did some SEM of that just to understand a little bit more about the morphological structures that allow them. They're basically like modified CD that are allowing them to stick those cuticles together, which was pretty neat. And I couldn't find any material of this in the Florida State collection of arthropods. So went to Beltsville, where the whitefly collection for the Smithsonian is held. And lo and behold, Louise Russell had collected this thing in Puerto Rico in the 50s. So it turns out we were like, oh my gosh, it's attacking this endangered thing in Fairchild Botanical Gardens. We need to be really careful we don't give it to Puerto Rico. Well, looks like it's probably endemic to Puerto Rico anyways. So that was a pretty neat find and made me feel better because Louise also couldn't decide what genus it goes in. She said Tetraleurodes, maybe Allura. We've decided maybe 4 different genera it could go in. We'll decide later. So just to kind of speedily wrap things up, data from imports and nursery inspections can provide a window into what we might expect in Florida. Having that reference material in the collection is really important for making those identifications. And the collections are really important for solving taxonomic problems when adventive species show up in Florida. I don't know why this duplicated pictures a bunch, but I'm always interested in scale insect bycatch, especially from Berlese funnels or turfgrass samples. And thank you to my collaborators. I'll take any questions. Yeah, good question. So the question was essentially, is the whitefly on the endangered Puerto Rican fern host specific to that endangered fern? And the answer is no. So it was also getting on other ferns in the same like greenhouses that they were rearing the spores from in Fairchild. So it seems to be a fern specialist, but it doesn't care what kind of ferns. And I think with finding Louise Russell's material from Puerto Rico, we have like over 10 different species of ferns now that it's recorded on. But it'd be great to go out to Puerto Rico and actually look at these wild populations. I think there's literally only 7 of this fern left in the wild. So, and these whiteflies are so insanely small. I'm not sure. We've kind of alerted Puerto Rico to it, but it's like, are you going to see them? Because it's almost like you have to tear apart the plant under a dissecting scope even to find them. So. That's a good question. So how does FDAX, DPI, and the USDA share data? So when we have like an inventive species arrive in Florida, we will alert the USDA, and the USDA will kind of like collaborate with FDAX, DPI to make decisions on whether they're going to like try to do an eradication program or something like that. But generally, it requires kind of like personalized collaborations to get your hands on USDA APHIS data, in my experience. But maybe some of the other DPI people can speak to that relationship. But yeah, I like Greg Evans, who works on parasitoids at the USDA, and Ian Stocks, like I have a close relationship with them. So the USDA APHIS data comes from them.
Our next speaker is Alfred, who is I guess from the Lourdes Nursery Research Center at Tennessee State University.
Good afternoon, everyone. I'm Alfred, a postdoc from Tennessee State University. I am here to present about our small collection and how our small collection as a whole is being used in tackling important agricultural problems. So a little bit of context here. Let's hear from our director.
I'm a professor of entomology and also director of Research Center here in Middle Tennessee. We have 5 faculty positions with Tennessee State University at our Research Center and also 2 USDA positions. My background in research is in ornamental entomology, and so over the years I've worked on a number of different pests, including invasive species like Japanese beetle scale, Japanese beetle, also a number of recurring beetles, the emergent beetles that are commonly affecting our nursery crops here.
Oh, thank you, Carla.
Hello, thank you.
Do you have anything more to say? Thank you again, Carla. Oh, here is Jason. You can hear from him as well.
University Nursery Research Center. My primary role at the Center is kind of management of nursery pests. The research collection we had here was begun at the center around 2001.
So it's a fairly new collection. Our collection started only at 2001, and I think Jason has to say. I think Jason has to say something more.
Yeah, the pest holdings that are some of the first documented finds in the state. Invasive pests have been recognized in our collection. We have a number of new state records, extension, and rediscoveries of insects at the time.
Oh, thank you, Jason. So here I'm projecting the important discoveries from our station. We have many rediscoveries. We have many state records, first records, and we have range extension, things like that, of which I like to highlight this European oak borer, which was first recorded from Michigan. But then we later found out that that was in our collection predating this first report. So let's hear from Jason again.
Charge for handling a lot of our specimen collections, our research associate here at the USF.
Oh, so who is this? Nadir? Nadir is here. He's very shy. So Nadir's dad is an entomologist, and he has been collecting insects from his very young age. And let's hear from him.
The person. Hello, my name is Nadir Youssef. I'm the insect curator at Tennessee State University in Powell, Tennessee. When I arrived at the University Research Center in 2002, the insect collection houses 12 drawers. At the present time, we have expanded that to 300 drawers. At the research center, I have two primary duties. One is to assist Dr. Alfred in his research of the insect pests of the nursery industry in Middle Tennessee. The second is curation of the insect collection. This takes up approximately 5% of the time.
Thank you, Nadir. So the real reason behind the expansion of our collection is Nadir. That's why I asked him to stand in front of our cabins and give him a talk. And so as he told that our collection is fairly new and we have 50,000 specimens in total housed in 12 cabinets in 300 drawers. And based on the nature of research that our collection is very much biased towards buprestids and cerambicids. So what is my role here in using this collection? So I'm working with Chrysobothus femerita. It's a species complex. There are at least 12 different members in this complex, and it is really, really hard to differentiate these 12 species. And when I say that, I mean it. This is the similarity between these members of Chrysobothus femerita. And you can see their abdomen, very beautiful, but very similar. So the only way to identify them is to kill them first and pull out the genitalia and look at and differentiate them. But we have a problem here. So this will leave us with no option that we cannot do any behavioral experiments using this specimen that requires identification of insects before conducting the experiments. So, so I'm using the spectrophotometer, which it is a non-destructive method. We don't have to destroy the specimens. Based on the collections available, I'm scanning them and trying to develop an artificial intelligent model or something to correlate that with the newly emerging beetle and identifying them before conducting the behavioral experiments. So this is the preliminary data that I got. So even the USDA taxonomist Josh Basham, he says that Chrysobothus femerita and Chrysobothus shawnii, the pink star, these are really hard to distinguish when they are alive. And using our spectrophotometer data, we are very hopeful that in the coming days, we'll come up with a model to identify them. I'm really interested in parasitoids, and I came to know that so there are many publications for parasitoids of Agrillus, but none for Chrysobothus. So actually, Nadir stored many parasitoids with a huge amount of natural history data on it, which triggered my interest to collect parasitoids. And all these were parasitoids reared from our rearing cages from Chrysobothus experiments. And so during the last spring, I kept around 10 cages. And I expected 1,000 beetles to conduct behavioral experiment, but this Phasconophora, it killed more than 600 beetles, and I got more than 600 Phasconophora in last spring. So we have a hope of conducting biocontrol experiments in the future using Phasconophora and other parasitoids, and also to publish these. So as our director told that we do a lot of Japanese maple scale research, and I noticed many emergence hole on it. And so we went for a survey, and bingo, the three new state records from Japanese maple scale. And these are the three species. Morletula prima, it's an exotic parasitoid. In 1975, it got detected in Maryland, and recently in 2022, Professor Woolley detected that in Texas, and in 2023, we got that in Tennessee. So we do a lot of Japanese beetle research, and Nadir collected a sarcophagid parasitoid that is really interesting. It's not taginid. And so last year we went for many surveys. We collected more than 3,000 beetles, and we break open the abdomen, and we were really hopeful that we'll get some flies. But to our surprise, we got a hyperparasitoid that is a perilampid. So I'm relatively new to this country, and at least that's the excuse that I use everywhere. So in Middle Tennessee, I had a conversation with a person, and with full enthusiasm, I started saying that what am I doing, why I'm collecting insect spiders and everything. So that person, after hearing all my speech, she told that while I'm going back to India, it is better if I collect all the spiders from Middle Tennessee because she don't want any spiders or insects in the entire state. So conducting outreach activities is very, very essential in Middle Tennessee. So let's hear from our technician what she have to do.
They do a bunch of shows with local kids at elementary schools and French clubs. And a few days of show, once the spiders, the kids really enjoy this. We also look at some other things, such as non-native insects from during the season, we can see what's much out for when we kind of don't want. Then we also show some symptoms of what these fears of these descriptions of spore damage that we can see from inside and outside. So the kids can just kind of see what insects do.
Thank you, Gabby. Meet our technician, other technician, Hank. Hank, can you introduce yourself, please?
Okay, good one. I'm leading the 3D modeling and printing project here.
3D modeling and printing, that's really interesting. Hank, do you use real specimens for your 3D modeling?
This is a real.
Oh, yes, the answer is yes.
I'm going to walk you through the process of converting this into a digital file. For the spray, we get a color comments so that the scanner will pick this up. I put it on here in front of the scanner. I put the thing. This scanner will now pick up the details of our specimen and convert it into a 3D model. That's good.
Using this digital file, we can make up anything that we want at any size. This is an example of a spotted lanternfly that Hank made, and this recently extended to Tennessee as well. And let's hear from Jason again. He may have to say something important.
We do have several concerns with this collection. These include things like the time we curate these specimens, lack of adequate system to track and catalog all of the specimens as well as digitize them. And unfortunately, because of that, our data is also not publicly available. So there's also a lack of resources to do that. If we really have room to expand our collection presently, our insects are housed at 3 different locations in the building, including even in the hallway. And our facilities are not really totally optimal for museum specimen storage with humidity and temperature control.
He's sad. I hope you all are sad. I wrote a grant and I got a reply that is more or less similar to Jacob P's reply that it's well written, but it's just a museum. With that, I end my presentation. I'm happy to take questions.
Your associate was spraying the ceramicin. Was that with spraying paint, or what was the substance they sprayed?
Actually, the question is that what is the substance, what is the chemical ingredient of the spray, the masking spray that is used over the 3D modeling? I'm really sorry, I don't know but I'll send you the details soon after reaching Tennessee. Thank you.
Any other questions? Thanks very much.
Thank you. Thank you.
All the way up. Next speaker is Jim Hayden. Florida State Collection of Arthropods.
We've got just a moment, so we can. Okay, thanks. Hey, good afternoon. My name is James Hayden. I'm the Lepidoptera identifier at the Florida State Collection of Arthropods. And I'm going to give a talk about some happy stories. Actually, most of these do not end in any kind of drama, to be clear. So, and first of all, most of these don't end in drama. And second of all, these are all involve moths. So if you want to leave and have a coffee break now, you're welcome to. Thanks, first of all, to Erin Powell for introducing the FSCA. That's a lot of what she said is, you'll understand from where we're coming from. Early detection of pests is our primary mission of the Florida State Collection of the FDEX DPI, of which the Florida State Collection of Arthropods is basically the reference collection, as well as that of other institutions in Florida, like the University of Florida and the elements of APHIS PPQ. As such, the FSCA serves a regulatory function. It serves to help us to identify what all the weird stuff that turns up in our state. So even if it's not a pest or not known to be a pest, people, you'd expect that people would naturally have, some concerns about things that are not novel, especially when they turn up in a place where there's no natural enemies or, novel host plants. This is just a screenshot of our regulatory database, the end place where when I receive a sample, I have to acknowledge that I received it and start entering my identification and all this and that. This just goes to show that, yes, we have to get things done in a timely fashion. If it's an interception, we have an hour. Things that are not like, things found out in an orchard or someplace, those need to get done, move it along. This is an example of the output that we have, one of the reports. So when something turns up that's a new continental record or something, we might not have a whole lot of time to find out whether it's, truly new to our state. We do the best that we can. So what happens when you combine an insect collection that has these regulatory functions where we have to produce answers that are quick, fast, and also simple? Like, is it a pest or is it not a pest? Simple answer, just don't think about it, yes or no, black and white. Combined with the fact that we are one of the largest insect collections in the country. Furthermore, the Lepidoptera is combined with is housed in the same building as the McGuire Center. So there are two collections, FSCA moths, the McGuire Center for butterflies. This is one of the largest LEP collections in the world. Misidentifications happen. This is an example of this is our part of our Noctuidae pro tem over here. So it results that often we have a lot of older misidentified specimens turn out to be, something that we just thought might be new. Here's an example of our photographs of our recently rehoused alcohol collection. It's in a former fumigation building. So I'm going to tell some stories about little anecdotes about things that have turned up. But the first one I'm going to tell is actually a story from the National Museum because Don Davis, who unfortunately passed away recently, if you didn't know, but he did the identification for this. This little Taniad moth is borzan bark, and it was not known to be a pest. It's native to the Caribbean, but about 20 years ago, it became a problem in nurseries in South Florida, where it kind of attacks the bark of seedlings and orchids and other, and also mulch. So what I've heard, and I heard this from Don, is I don't know the whole story, but they had somebody tried to sue somebody else because somebody is, one company thought that another one brought in infested material. So Don went into his collection in the USNM and found out there was a few specimens dating back to the 1950s in that area and into the 60s and 70s, 80s. So at that point, the prosecution's case fell apart because you can't really make it prove that this did not come in from the environment. Things might have just flown in through the window of the nursery. We also have some specimens of the same lot also collected by Wolf and Barger in our collection. I found after looking into this. Now here's one that directly affected us. Palpita persimilis. This is this nice little crambid moth. Is probably the worst pest of olives in South America, where it's native, where olives are grown, olive foliage. We first detected it in 2012 in Central Florida, where a master gardener, her Japanese privet was being infested by something, and being a good master gardener, she did not immediately destroy it. She contacted the university, which contacted us. What is this? Let's find out about it. Well, I dissected these things because you have to dissect these moths with no wing pattern. And it was not one of the native things. It was this pest. But then I went back in the collection and found that, yeah, we've had specimens going back to the 1980s, back to 1980 in the Fort Lauderdale area. And we had a few records here and there. Yeah, it seems to consistently attack Japanese privet, Ligustrum japonicum, and a couple other natives. We have a couple of records on our native olive, our nascent olive industry. But this gives us kind of a track record of what this thing is doing, has been doing for 40 years. Here's another species, Homoledra howardi, which broke out as a pest of coconut, this little moth, coconut foliage in Fort Lauderdale area about in the 1990s, where a UF professor Bill Howard made a big deal of it, justifiably so. It's pretty nasty. I described this thing about many years later because nobody else was going to do it, honestly. And we had most of the specimens. So, but in the process of working out the diagnostics, I found ketotaxic differences in the larvae, which allowed me to go back in our collection and found that, yeah, we had specimens of this thing collected on other palms from the 1970s onward. Maruca vitrata is the bean pod borer, is a huge pest of legumes in the tropics. It's a pantropical distribution. It's also in the Caribbean. In 2018, some amateur collectors in the Florida Keys collected a specimen and sent it to us. They recognized its importance, and we organized an emergency survey in Big Pine Key in the Florida Keys for a week. And we didn't find any more, but in the preparation for this, I knew this had been collected once before in 1980 by a great amateur collector, Terry Dickel. This man is he's still alive, lives in Central Florida, but he spent years collecting in the Florida Keys. And when you first think that you might have a new record from the state of Florida, go back into Terry's collection, because he probably got the first one back in like 1976. In this case, 1980. I visited Terry in preparation for this. He had like 10 more specimens. He just didn't recognize the significance of this, but they were collecting about once a year or so. And then nothing more after that point. Why? It could have been because Terry moved up to Central Florida, or maybe there were changes in Cuban agriculture in the 1990s. We don't know, it's speculation. But it gives us a track record that this thing is just turns up, but is not establishing in Florida. This species didn't involve our collections, but it's of interest because everybody is cultivating hemp now. Graphilida delaniana was the hemp budworm, is a huge pest of hemp. It's a Eurasian species that was first identified in 1982 by Bill Miller at the University of Minnesota. He went back in other collections and found that, yeah, this has been it had been in the US since the 1940s in various states. It's just that nobody was really doing any surveys on hemp at the time because it was not really a cultivated crop. Now it is. So it's kind of a good idea to, collect things on plants that might not be important at the time, but somebody might start cultivating that thing in the future. And now you have a record of what's, of their natural their enemies. Here's an interesting case, a blastobasid moth. These are usually not problems, but this is a species that moves around a lot. In 2017, I got one of these was intercepted on a shipment of palm seeds from Florida into California. The California's CDFA barcodes everything. So they barcoded this caterpillar. And the next thing I know, Mark Epstein gave me a call saying, "Hey, do you know this thing is in Florida?" And I said, "Well, it's news to me," but I found a picture of the genitalia, which I routinely dissect and published in another work. And I said, "Hey, this thing this is just the same as a few other slides that I had just made over the past couple of years of stuff that turns up in suction traps." So great. So it's been here for a few years, and it doesn't seem to be moving north, but So this is a good reason to kind of identify stuff and keep things, identify things even if we don't think they're important. Dryadala terpsichorella, this is a bit of a nuisance pest, another thing that moves around, and occasionally it mainly feeds in fungus, but it can scrape, in no choice tests, it can like scrape the epidermis of green plants. It was initially reported in 2008, but a few specimens I found in the collection were actually collected about 4 years before then. This is a good reason why too, if you throw, we throw out specimens all the time, okay? We think there's something, but keep a few specimens, just keep a few. If you think it's common, it's unambiguous, hey, I could be misidentifying things. So I pin a specimen once in a while and my successor could correct me. Here's another thing that turned up in 2007. This is a bit more dramatic. It's a scavenger moth that tends to get into, it can be mistaken for, it has a habit of getting into rotten old cotton bolls in the old world and be mistaken for pink ballworm, which is a primary pest of cotton. Yeah, we do have a relictual pectinophora infestations in Florida, in particular parks. That's a whole story by itself. Yeah, this thing actually accounts for a few misidentifications of pectinophora elsewhere. It turns out that this thing turned up in Florida back in 1975, based on pinned specimens in our collection. And it probably spread around the state because it likes to get into cattails. It's one of those things that makes cattails fluffy by getting into the seeds. So, Florida, 1975, Texas, 1983. So we have an idea of where this is and what. So I'm going to conclude with a few recommendations. First, we all know, sort the collections. Yeah, right. Do we have time for that? This is, if you can't sort everything, search through the things that are, your jurisdiction, prioritize the state that you live in. Does it occur in Florida or not? That's all we care about. That's one of the first questions we care about. Focus on the earliest specimens in your collection. Earliest collected ones in the FSCA are like a few from the 20s, but mainly things got going in the 1950s. Hey, if you have returned loans, sometimes the expert keeps the good, important, weird, interesting novelties until last, and some of those things, interesting new records, might turn up in their returned loans. Always ask for real specimens. This is the age of AI. People like to send fake photos to us. And you can't really, a lot of these things you have to cook or sequence anyway. If you're bopping around Florida and you think you could collect a pest, please talk with us. Would like to find out more, and we'll keep the information on, sub rosa until, the authorities decide it's okay to release. But with that, I'd like to acknowledge a bunch of people, Paul Skelly and Eileen Buss, for corralling a lot of these records and information into a large forthcoming publication. All the collectors, Terry Dickel, Lyle Buss, David Fine, Jim Turbridge, Julieta Brambilla, and also Sidney Bennett, Charlie Cavell, and Liz Soma, and many other people have helped to sort this stuff in our collection. Okay. Thank you.
What I will get my turn over for adult moths. Yeah, as I said, how do you deal with
How do we deal with identifying caterpillars when we have a limited deadline? It's not a firm deadline, but there's a truck waiting at the border. People want answers. I might identify things to family and just say, I'm sorry, we can't. Unlike California, we have, California can, from what I understand, can turn back things that are not known. We can't. If I can say, okay, it's a blastobacid larva, family is established in Florida, probably not a pest, they'll probably let it in, honestly. And then maybe they'll do a follow-up, okay? If later on, I look at it again, we sequence it, then one of our inspectors can go to the grocery store to which the shipment ended and do a follow-up inspection there. But yeah, we don't have, if I don't have time to get it down to species, then we just have to say family, genus, shrug. Thank you.
Yeah. Okay, thank you. So short. My name is Lourdes Chamorro, and I'm a research entomologist with the USDA's research arm. And I specialize on weevils. And we also have, just like James mentioned, we also have this 1 hour turnaround, more or less. It's up in the top there. We also get weevils sent to us, and we have to provide this identification. But first of all, I want to thank Jessa Watt and Luke Kreslin for inviting me to present my work. And when I was first invited, I was considering how would I talk about this. I could talk about some of the weevil work that I've been doing in support of U.S. agriculture. But you can't swing a dead cat without actually hitting like an agriculturally important weevil. So this is just like a super difficult task to do. I have done a lot of research on weevils, some of the dealing with association of larvae in adults, to also determining identity of difficult to identify weevils that are intercepted, say, with avocado, and some of that has been published already. So there are endless examples of what I do on a daily basis. So I decided to instead talk about my current research on Agrilis with Kojun Konda and Greg Gustafson and the acquisition of the Edward Yendig collection of Buprestidae. So in 2002, several species of ash tree in the United States and in Canada began to present a characteristic sort of dieback at the crown of the tree and sort of this epicormic kind of growth at the trunk. And the beetle was found to be a buprestid, and the only person in the world who could identify this beetle was Edward Yendig, who was sitting in Slovakia. This was Agrilis planipennis, the emerald ash borer, and it belongs, so this is the beetle, and it belongs to Buprestidae, and it contains the genus more than 3,600 species, and possibly more than 2,000 remain to be described. And this is according to some of the work that Edo Yendig has done. So the species is native to East Asia. And in its native range, it causes minor damage to weakened or dying trees. But in the introduced range, it attacks and kills healthy trees in the genus Fraxinus or ash. And it's one of the most devastating insect pests in North America. And it has destroyed more than millions of ash trees, costing billions of dollars. It has also moved on to a new host, the white fringe tree in the US, and it has been confirmed to feed on olive trees. So this is a very significant pest. And since its discovery in 2002, it has since spread throughout the eastern United States and Canada. And it has been found as far west as Colorado. And in 2022, it was discovered in Oregon as well. And the demise of ash trees in North America is also impacting cultural practices of North American indigenous communities because they rely on the bark of ash trees to create these really beautiful baskets. And so that's being impacted as well. So we have a problem with this gigantic genus where we don't really understand the relationships of the species, what is potentially invasive, what's related to the emerald ash borer. These are some of the questions that we have. We can't really make predictions about the emerald ash borer and other species that have similar evolutionary histories. And the classification is really confusing. And right now, 37 subgenera are recognized. And US workers have not really adopted this subgeneric classification, and it's sort of applied sort of piecemeal across the world. And in 2011, Yendig, Edward and Grebennikov, they started to designate these species groups in order to make it a little bit easier to identify these 3,600 species, right? So it's really difficult. And they published this really important work on the Agralis of East Asia. And in 2015, I also was working with Edward Yendek and several people from the US and Canada in order to determine, okay, what's related to the emerald ash borer? Does it actually belong in the subgenus Eurygrylus? And we concluded that it actually is closest to, but this is based on morphology and some of the work that Edward Yendek was doing, that it's related to these East Asian Agralis species, the cyan and Niger, what we call, I think, I forget what the species group name is, Pseudolubopretzery and so on. And in 2019, the Canadians and the Europeans used 3 gene fragments to infer the first phylogeny of Agralis. But a lot of their work was based on very limited regional sampling, and it included 90 species. But what they did find was that there was non-monophyly of the subgenera and species groups. So this is a really consequential group. Not only is the emerald ash borer an invasive species in the United States and costing millions of dollars in cost to replace and treat all of these infected trees. And we lack but we also have 12 species as well in the United States, additional, that have been accidentally introduced. And some of, I think one of them wasn't actually mentioned earlier. And we lack a phylogenetic framework for making predictions about future incursions, and this limits our ability to effectively respond and control future pests. And aside from that, we are facing a decline in taxonomic expertise. So Edward Yendek is retired, and Henry Hespendite is also retired. And taxonomy is really important for trade policy. So if a species is native to the United States or not, that has very direct trade policy implications regarding international commerce. So this study that I'll be talking about was made possible largely by 2 factors. So this improved technology for extracting and sequencing DNA from degraded samples from museum specimens, and the acquisition of the Edward Yendek collection of Agralis. So maybe now 5 years ago, I spearheaded an effort to acquire the collection, Edward Yendek's collection. So he basically says, I'm retiring and I would like my collection to go to the USNM. And we were able to get buy-in from major agencies in the US government, which are listed over here. And we acquired the Edward Yendek collection. This is an invaluable reference for the identification of Agralis, and it contains more than 25,000 specimens of Buprestids, about a quarter of the species. And it's a dense sampling of Palearctic and Indo-Malayan fauna. And this is 2 decades of Edward's life that he spent trying to put names on his collection and material that he had in his collection, authoritatively identified. And a lot of it too was recently collected in places that probably no longer exist. So the specimens and the way that Edward curated his collection is very unique. So it's sort of like this in-between European sort of US way. You really need to see the underside of these Agralis. So he has this L-shaped card that he uses, and he also dissected the specimens, which were just beautifully meticulously spread. And so this is great to be able to identify these species, these Agralis species, but it seems that that might not actually be the best for DNA. So we'll talk a little bit about that. Okay, so we have now this conversions of people, Code and Con, that had just moved into our lab. We have the Edward Yendek collection. We have this new technology, new analytical methods. And we submitted a proposal to the US Forest Service international programs to basically infer the first global phylogeny of Agralis with including as many specimens as we could, drawing from this collection from this new acquisition. examining host plants across the genus, and providing a foundational data for molecular diagnostic development. And then to put all of this information into a centralized resource for taxonomy and biology. And so today I'll be presenting some of the preliminary results of what we've been doing. So a lot of this is Cogent Conda's work. We were very ambitious, but I think that's the only way that you can actually deal with a genus that is so diverse. So our ultimate goal was to include around 800 species. And we had a lot to draw from the Edward Yenda collection. So we were trying to also do our DNA extractions so that it was non-destructive. So this is going to get a little bit technical here, but okay, just bear with me. So we didn't want to increase the degradation of the DNA. So we took care also to minimize the contamination and the entire extraction process. We really took care not to sterilize everything that we were using, our surfaces, and the triple, double bleach solution to not cross-contaminate. So I guess pretty standard wet lab protocols. So what we discovered was that not what we were expecting. So what we were expecting was what we're showing here in the graph, where we have a lot of fragments still in the thousands of base pair range. But what we actually got was sort of median length with about 80 base pairs. So what we're seeing is that the yield was very low and the DNA was degraded, and that's probably due because of the way that he was curating his collection. So now what we're doing is, instead of looking for the specimens that are like just really beautiful with the legs all spread out, we are looking for the ones that look more like the ones at the bottom there. So, we're going to show some of the results, and this is, so let me go back a little bit. So because of what we were facing with DNA recovery, so how do we sequence these specimens? So we did shotgun sequencing, and we did recover a lot of the mitogenome and ribosomal complexes, even for specimens that had highly fragmented DNA. And we were, so Cogent did, in silico simulations, and we found that we couldn't really use the Coleoptera probe set. But we were able to generate, and I think what appears to be a very consistent results overall, with 6 mitochondrial genes and 28S at 9,000 characters. So I'm not expecting you to read this. This is very preliminary, and we're going to continue to add more data. This is from 165 terminal taxon, but we are going to, this is just 1/4 of what we're going to be adding. So yeah, the subgenera are not monophyletic, so we probably need to redefine some of the groups. We included some of the other genera within the agrolines, and we have been finding a lot of them within Agrylus. So I don't know what's going to mean. It's like Agrylus is going to continue to grow, kind of like Staphylinus. So Agrylus planipennis is recovered, where we were hypothesizing with the sign and anger group, but we'll see where this falls out. And we have a large mitogenomic data set, including the CO1 barcode region. This takes us to the next step of this very ambitious project, which is to develop a centralized resource for Agrylus taxonomy and biology. And we are building this with TaxonWorks, through TaxonWorks, to build an integrated web-based workbench, which will include nomenclature, literature, distributional data, published host associations, and we're also digitizing all of the types from the USNM, as well as the ones that we just acquired from the Edo Yenda collection, and this molecular data. And these are some very beautiful images of the more than 600 types, primary types, and keys as well. So this is just to the US and Canada species, and these will be made publicly available. And of course, we look for collaborators. Please contact Code and Conduct for that. And we'll be expanding to include specimens from the Afrotropical, Australasian, and Neotropical regions, including the Corletti collection. And then just one final thing. This entire project was made possible because of the work that Edo Yandek did for 2 decades, trying to compare his material to type specimens. And just something to leave out there, and this is something that my husband, Matt Buffington, and I have been talking about, is the prioritization of the digitization of types. And this is if I had a little soapbox, I would get up and I would say, for those of us who are here, who are curators, who are collection managers, why are we not prioritizing the digitization types? And I'll leave it at that. And here is my acknowledgement slide. I would very much like to thank our funding agency, USDA, and the U.S. Forest Service. Thank you. Well, you can catch me later.
Good afternoon. I'm a little anxious here to follow my spouse and be right before the next speaker, John Heraty. So I find myself in a unique position, but I'm greatly honored to be here. And this is the crowd I love to talk to. Some of this is going to be a little probably stuff you already know. I'm trying to give you maybe a different way to think about it. I like to think of collections as almost like time machines, and they allow us to go back in time, see things, see observations, see people's notes. And as custodians and curators, we're the ones keeping track of this stuff well into the future. I'm going to give you 2 examples where all of this kind of came together in 2 agricultural related systems. These are the 2 taxa that we're going to talk about today. One's a Celeonid, Trisulcus japonicus. The other's a Fagitid, Leptoplena japonica. We'll start with Trisulcus. These are pentatomid egg parasitoids. We became interested in these because they were the primary things attacking the brown marmorated stink bug when it was first emerging in the United States around 2003, 2004. Sorry, this is on some sort of auto advance. I'm going to have to keep going back a little bit. So Trisulcus was reared. It was sent to me for identification. I was able to get to genus real quick, and we had the name Haliomorphi for the species. I sensed I was out of my depth on this one, being a Fagitid expert. So I was able to convince ARS to hire a postdoc, Elijah Talamus. And within 3 weeks, Elijah was able to determine what people were calling Haliomorphi was actually Trisulcus japonicus, and the name was well over 100 years old. The type specimen was sitting outside my office. We had 2 other specimens in the collection, and that was it. He was able to tie all this together, and we very early on in the process had a stable taxonomy to base our foreign exploration on. This comes into play here because his advisor, Norm Johnson, revised Trisulcus for North America. He used the USNM collection and every collection there was in the United States, recurated them, and provided a stable taxonomy, establishing what we call taxonomic preparedness for the situation to come. And this was the situation to come. Trisulcus japonicus suddenly showed up in the United States on its own. It was in quarantine where we were doing host range testing, but it showed up on its own. We were able to know this because 1, the taxonomy was already worked out by Norm Johnson, and 2, the taxonomy of Japonicus was worked out because Elijah had the wherewithal to look at the types and put the 2 and 2 together. From that point on, we knew what we were working with, and that helps us with workshops. It helps us get the biocontrol community trained properly so they know what they're looking for. Okay, that's kind of example 1. Here's the next one. This is the one I've been working on for the past 6 years or so. On the right is a spotted wing Drosophila. On left is its enemy, Leptopolina. That's probably Leptopolina japonica right there. This is this Drosophila is a pest of saw fruit, and it's a serious pest, very expensive pest. My former advisor, Johan Nordlander, worked out Leptopolina nomenclature and taxonomy in the early 80s and kind of left it at that. A number of new species were described since then, but the stability of Leptopolina was never in question, and the species limits fairly well worked out. Eventually, my student, Xiaohua Liu, worked out the species limits of North American Leptopolina and did actually an extensive survey of Leptopolina of North America. And this happened to be just perfectly timed for the arrival, we didn't know it at the time, but the arrival of spotted wing Drosophila and its natural enemies. These are two that we deal with on a regular basis, surveyed across the United States, Leptopolina japonica and Leptopolina heterotoma. In the case of heterotoma, this one was already known to be in the United States, and we have several specimens in the USNM and around the country. Japonica was my mistake. I mistakenly identified Japonica as heterotoma in 2016, but I carefully put those specimens away in the collection. Later, when it started to be revealed that this thing, heterotoma, appeared to be polyphagous because it was attacking not only Drosophila melanogaster, but also spotted wing Drosophila, I went back to the specimens. We started doing a little bit of sequencing. I started doing a little bit of morphological work. And as it turns out, Leptopolina japonica was already in the United States. It wasn't just in quarantine where it was being tested for host range. The DNA was able to help separate it. And then I went back and looked at the scutellar morphology. And sure enough, they're pretty easy to tell apart now. But that was not the case when this was all taking place. So I want to look at two different scenarios if this didn't work out the way it did. Scenario is a pest arrives, it starts playing havoc, and people freak out, okay? We all start doing surveys. Well, what's attacking it? What's killing it in the United States? If those identifications are not done authoritatively or done thoroughly, and in many cases, what I deal with are non-authoritative DNA barcode data sets on GenBank, where things are sequenced and people get a name and they're assuming, and they have the right to assume, that they're getting the right answer. But these databases are often populated by erroneous records or non-authoritative records. The conclusion in this case, in both the Trisulcus case and Leptopolina, if you didn't know that the parasitoid was actually native to Asia, you would have concluded the natural enemy was actually already in the United States and was already doing its job. Whoa, it's already here. That's great. But as it turns out, that would have been an erroneous claim to make without being able to connect it to the actual origins of the parasitoids, which happen to be in the same area as the pest that they're attacking. This would lead us to missing some pretty critical pieces of information here. One, and this is actually one of the most critical ones from a regulatory standpoint, is we would miss the fact that there's something non-regulated actually out there already doing the job. That actually changes considerably how host range testing is done and how foreign exploration is done. So that's costing us a lot of money. It might be considered unnecessary to do host range testing or go into foreign territory to go look for natural enemies because we'd already have native ones doing the job for us. The one from an evolutionary standpoint that I find actually most interesting is we'd actually misunderstand host ranges. The fact that Leptopolina heterotoma, which is a well-known Drosophila melanogaster parasitoid, it'd be very interesting to conclude suddenly it's competent on spotted wing Drosophila, a species it's never seen before. That would actually change our interpretation of how parasitoids even evolve and what their host restrictions might be. This would be, this results in misunderstood sources of biological control, which is critical. And we'd also miss an opportunity to see the spread of a parasitoid, an actual laboratory experiment on a continent-wide range. We would miss that entirely because we wouldn't have the expertise to know we were actually looking at an Asian species, not a North American species. Together, these intertwine between kind of a triumvirate of biological control, taxonomy, and collections. Those three things have to come together for really effective classical biological control. If there was no sound taxonomy by the people before me that were really looking at these wasps because they were just surely curious about their taxonomy, if that wasn't done, then we wouldn't have the revisions, we wouldn't have the proper taxonomy to actually put names on them. And we wouldn't actually have any of the well-curated collections because we wouldn't be able to actually identify anything. Together, this would have actually impeded because we wouldn't actually have anything to go look at for me to compare, and we wouldn't have the types in the first place. That's pretty much all I've got. It was just a quick example of how two different species, within a relatively short amount of time, we actually got the identifications right and hopefully avoided some serious mistakes. I'd like to thank these people, and I think I have plenty of time for questions. Thank you, sir.
I'm interested in your opinion. Very easy to say what you say when you have agricultural interesting taxa. How do you defend the same thing versus for me when I need to apply that this is I'm going to make comments when these are not exactly one-to-one connected to agriculture.
Okay, so
how can we make this?
okay, so the question is, how do you make an argument with respect to economic impact of a perhaps non-economically important insect? I would challenge everyone in the room to consider, how do you know what you're working on is not going to have an economic impact? Because I tell you what, Norm Johnson he revised Trisulcus because he thought they looked adorable. There was nothing more in it than that, and he was fascinated by them. I don't know why Nordlander picked Leptoplena. That's the last genus I would pick, but he did it, and he did it thoroughly. None of us know, none of us have the crystal ball. If you do, please share it with everyone, because then we could probably save ourselves a lot of time, but we don't know. We don't know what they're going to be. So it's best to have as solid taxonomic preparedness as possible, and that means kind of covering all the bases. This one's going to be tough, I can tell.
Well, out of curiosity, I have a question. Are there, besides foods, are there things you know she's doing this?
With these two cases, perhaps with stink bug, I know because it's a household pest of an enormous proportion, not just a nuisance. That one absolutely. Spotted wing Drosophila appears to be an agricultural pest, and I don't know of any human impacts as of right now. Yep. That's right. Yeah. Yeah, so just repeat her comment, we talked at lunch about, basically why, if we kind of know more or less where most of the types are of the whole metabolous insects of the world, like why haven't we digitized them? There's a number of reasons why. It's a very complicated answer, I have a feeling. If we can critically come together as a community and give very strong evidence why this would be a very high priority research item, not just a waste of taxpayer money, I think we would be way on down the road here. I'm interested in linking a definitive barcode sequence to an actual vouchered specimen that actually has been identified by an expert. To me, that's, to use a term that John Heraty came up with, we call that a DNA holotype, or having that linkage, I think, is absolutely essential. We're now in the point where we can actually be sequencing the holotypes themselves, sometimes upwards of 100 years old. And I think we're possibly entering a new phase of the relevance here of taxonomy. And I think finding these types, getting them digitized, making them aware to the world, it's our job as taxonomists to tell these stories. These insects can't speak for themselves. Sometimes they're of agricultural importance, whether good guys or bad guys. Sometimes they're just beautiful to look at. But as long as they remain kind of in the dark like that, that's going to be, we're all kind of missing out, I think. So this is just, I'm hoping, one example that you can kind of take home and appreciate. Great, thank you.
Last year, I was exposing Chamorro from the University of California.
Thank you. They bring on the old guy last to make everybody else look good. Okay, so this is a little bit outside of my sphere of influence. It's not eukaryotes and things like that, but I'm going to be telling you a story about, largely about vouchers and the importance of vouchers and how they're fitting into things. And I think, you know, that I came up with this weird title based on Back to the Future. The commonality I have is both Michael J. Fox and I are both Canadians, but other than that, it's not there. But collections are really a way of walking back in the past. We can always go back in the past and we know exactly what that specimen looks like that somebody mounted up at that particular time, and we can use that. So I hope to give you some examples in here of what we're looking at here. The second slide over is looking at the citrus experiment station. This is still on our campus. It's owned by the business school now. Citrus was a big thing in California. It was a big economic crop, and it was where all of the research was being basically focused on to try and resolve citrus issues as they were coming up. And Harry Scott Smith was one of the people who was instrumental in building and directing that first citrus experiment station that eventually became the University of California, Riverside. This is our current museum. Just to give you an idea, it's there. It's a nice, pretty one. This is actually an old picture. And we, of course, have our collections in there, and there are all these big things. And everybody in my lab knows that if you can put a pin through something, it's not really worth keeping, but that's what we have to deal with right now. And that's not what I'm going to be talking about. I'm going to be talking about my favorite group, which are the little guys. So this is your American dime with all the little calcids on it. So that's going to be my focus of interest, because that's also the focus of what the people worked on initially in those collections. I like to call this the biocontrol rat pack. Some of you may recognize the names of people there. I love the one in the bottom right there of Paul DeBach, smoking his little cigar, sitting around with his t-shirt way back when. But you had Harry Smith, Harold Compeard, Paul DeBach, Stan Flanders, Ted Fisher, Paul Timberlake, except for maybe Timberlake a little bit. They're all involved with biocontrol. They're ecologists out there in the field trying to resolve economic problems. They weren't really taxonomists, but they did describe chalcidoids. So here you have biocontrol researchers that are out there doing there. I love Harold Compeard, 240 species that he described. And he was traveling around the world to collect things to bring back to Riverside to release. Then we were followed up by Curtis Clausen. He didn't do well here, but you know, he obviously contributed great things on behavior. Gordon Gord came in, described 47 chalcidoids. John Pinto, he came in as a Maloid person. Earl Oatman, a biologist there, forced him into working on trichogrammatids. He did get 90 species, and we'll talk a little bit more about that group later on. We then have Sergei Trapitsyn, who's here in the room. He's one of our principal museum scientists. He's outscored everybody in terms of the number of species that he's collected. Amir Me, along with students, 199 species. We then branched out at UCR to a new taxonomist. We have Christiana Weirauch. So far, she has not described a single chalcidoid. I don't know what's wrong. Anyway, I want to return back to the collection here. So Harry Scott Smith was, this person was responsible in 1923 for moving specimens down from Sacramento to make this the first building of our actual collection within the citrus experiment station. The insect quarantine facilities were central to all of this. Things were coming in from around the world, and they were using them. They were researchers that, unlike most of our researchers we have today in our collections, believed in the value of vouchers, of putting them in the collection, maintaining that historical record. Our current museum here, again, built in 1994, we've got 4 million specimens, we've got a compactor system, and we've got a pollinator garden. That's also a newer picture. And again, I'm not going to talk about these old specimens. What I'm going to focus in instead here are the slides. The slide collections largely what was developed by these earlier researchers in our collections.
And of course, they're wonderful specimens. Look at those slides. They have such wonderful data. We even have some database things on them. Look at this one, collected by C.P. Clausen. Look at the massive volumes of information that are available on that slide. And at least it's in Canada Balsam. So we can at least accept that one as being a fairly good one. But because they were biocontrol researchers, they did something, and we were lucky because nobody threw them out. We have cards that go along with a lot of those collections. So there were numbering systems associated. So a lot of that data could go back and be retrieved that were there. So there were ecologists, they weren't taxonomists and people interested in making nice little specimens, but they at least did capture that data. So now we have our main slide collection, and we have a pretty, when I summarized it for myself, I was astounded. 80,000 slides of trichogrammatids, all of those pretty much developed by John Pinto, all because of a goading by an ecologist, Earl Oatman. We have aphelinids, 25,000 slides, of which we have a lot of them database. We have 16,000 mimerids, that's all largely Sergei. We have 18,000 insertids, that's mostly Gordon Gourd. We also have 18,000 Thysonoptera and 16,000 mites. So that's more than 170,000 slides in our collection. That little thing on curation grants below, a lot of those slides were originally mounted in Hoyer's medium. A lot of you know how bad that was. So we had some nice curation grants to go through and turn these into real specimens. That little person there by, I think it was Marlene Plantuine. She was one person that remounted a lot of these slides all as a single individual, and we really appreciate her work. So why are voucher collections good? Why do we care about all these vouchers in our museums? I give some examples from Sergei. If you want to get the true details on this, ask it from Sergei, because I get a little bit muddled when I start talking about somebody else's research. But I think Sergei's, a lot of his stuff is relevant to how we're using our collections to go out and discover things. So he's untangling the Anagyrus pseudocossi complex. He's using a combination of both molecular methods and morphological methods. We have one here, a new description of a male of this other species down here that was important for biological control. The last one I really thought was really cool was basically he was able to sequence two different things. They described a new species. The primary morphological difference was a little discoloration of the antenna at the base of it, whether it was darker or lighter. And they were able to actually document that one species was originally released into California. It was then another species came in and then replaced it in California. So you're able to use those voucher collections, just the slide mounted things that you don't have molecular data for, to be able to resolve that question. Sergei will probably think I'm saying it all wrong. But again, why do good collections make good stories? And this is my story. So this comes in with, we have a recent phylogeny now, and I'm going to talk about the family Calicidae, which used to be Aphelinidae. So we're trying to resolve the taxonomy of that. You've seen maybe our recent papers that we've had. So Calicidae is based on the genus Cales. We have one species which is found in South America, 3 species which are found in Australia and New Zealand. The one species from South America was described as Cales nowaki, and it was released around the world because it's a wonderful biological control agent of woolly whiteflies. So initially, a long time ago, we got involved in molecular systematics projects, and we were looking at phylogenies, and we needed to include Cales. We had 2 students, James Monroe and Youngwook Kim. They went to our biological control grove, which are on campus, and they collected Cales from one row of lime trees. So these are on the same row of lime trees. They got different individuals that were so different in their 18S and 28S DNA that we thought they were different genera, potentially even different subfamilies. They were vastly different. What's wrong? They kept blaming each other. Your sequencing was bad. Your sequencing was bad. No, your sequencing bad. So how do we resolve that? We go out and sequence a bunch more specimens. And when you look at this and you say, okay, from the same trees, we didn't go to any other trees, we find 2 different main groups. One of these we were able to identify as Cales nowaki, and it does basically also include its matches to specimens that were collected from Italy and from Chile. These Cales nowaki that we have were part of a biocontrol program where they were going to southern South America and bringing specimens back to UCR and releasing them against the whiteflies. The other species, we only have it from UCR campus, nowhere else. Where did it come from? How did it get established on our particular campus? And they're obviously not coming together. Males are really cute. They follow along the females and they have these real puffy antennae. And then when they come up against a female, they go poof, poof. And basically chemical recognition is probably big, which is why they're separating themselves out on those particular lime trees. Okay, what about all the museum voucher material, which we cannot get DNA from? What are we going to do with it? And you can see those wonderful biocontrol researcher slides there. Every single slide with a single individual. No, they used a pepper shaker and just sort of dropped them on the slides. They just wanted to have a lot of vouchers so that they keep track of these things. So how can we go back and use these? And here's Jason Modern, my PhD student at the time. He came in and he decided that he's all doing this whole part of this project, and he did four-wing geomorphometrics, because these are species which you can't just look at them and say they're different species. They're nice, little, beautiful, cryptic species complex. But he was able to find some wing characteristics that were useful in separating them. And he was able to go through these voucher slides to be able to sort out that particular story. And he ended up with Caly's nowaki, and we have another species, Caly's rosei, which was described. So, and what happened was the original importations that came to California, we can go back to those slides and say they came from Chile and they came from Argentina, from two different locations. And you could track in and say that, okay, we brought two things into California from Southern South America, but only one of those was exported to other areas around the world. So again, this is that example of where you can use those museum hard collections to help you to pull that history back and answer a current relevant question. So when I talked back to the past, good collections to make good stories, UCR collections were first established by biocontrol workers, not taxonomists, not anybody interested in systematics or anything like that. Collections were first intended as vouchers for research studies. That was their primary goal. Trips around the world for agents, especially by Harold Compeer, expanded our general collection. And our collection is now housing this monstrous 175,000 slides, and even slides with no DNA can be useful in making new discoveries. And I want to thank all the people that have contributed at all sorts of various periods to parasitoid slides in the UCR collection. I think they were amazing in what they did. And some of them just, like John Pinto, just the number of trichogrammatid slides he's put in there is absolutely amazing. But everybody's contributed in various ways. This is my advertisement. I have little cards for you. We have a new book on Chalcidoidea, and we also have a new database for that. You can take pictures. I have cards. I have little cards around if you want those. And also, I know you've all got this. Nobody's yet bid on this book. I'm looking for ways to raise money. I'm trying this out. It's a first edition of Dobzhansky's Genetics and the Origin of Species. It's got barely any markup in it, just like a signature line on it that's for the person that owned it. And this is a good way to donate to the Graduate Student Endowment. Get yourself a first edition copy of a book. You gotta have great book collectors out there that are just looking for this. So use that QR code. It'll tell you how to make donations and everything like that. So with that, thank you. It was not a fun process. I mean, Sergey's probably got more details on that than that he could expose. I mean, I believe there was a paper written up on how we actually were able to do it. So basically, I mean, Hoyer's is a liquid medium, and it's dissolvable in water. So you can, it was basically dissolving the specimens, and I wish I could, I really can't focus on her name, where it was, I think it was Mariana. She was able to, you transferring over to Canada balsam, and you go through the same washes of dehydrating it and running it into Canada balsam. And we got back to some really excellent specimens. And that was really focused on making sure that we had all the types taken care of. Some of the types were on black slides that you could not see anything into them until you actually floated off the cover slip and got a better look at what was underneath that. But the specimens all held together pretty much fairly well. Must be coffee time. Yeah.